So we're going to now do a self-cross on that F1 generation, and here's big S, big Y, crossed with itself, big S, big Y, and we need to work out the possible combinations of gametes on here. Now, from meiosis, hopefully you'll remember that there is something called independent assortment at metaphase 1 and 2. If not, that's coming up next. So what we're going to do is take random alleles from here, and the rule is you can have one of each letter. So let's give a bit of space and let's work out this Punnett square. We have two parents, big S, big Y, and the alleles that you can do are big S, big Y, that would be this one and this one. You can do big S, y, which is the first one and the last one. You could do little s, big Y, and little s, y. Those are the four possible different combinations of alleles if you follow the rule that you can have one of each letter, or one allele of each gene. As we're crossing it with itself, it's exactly the same up here. Same combination of alleles. And all you need to do is add all those letters together. Double dominant there, and so on and so on. I'll save you the pain of going through it all here. And what you'll end up with is a number of traits. So let's fill all this in. So there we have a completed Punnett square for a dihybrid cross, looking at two genes. In this case, smooth and wrinkled represented by the S alleles, and yellow and green represented by the Y alleles. And here are the 16 possible combination of alleles that you can get. So now let's see what we can do with some phenotypes. If there's anything where it's dominant, big S something, big Y something, well, we find that there's 9 of those. There's this one, 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 1, 2, 3, 4, 5, 6, 7, 8, 9. So there's 9 of those guys, dominant for both traits. Now if we look at, say, dominant for smooth and recessive for color, we find that there's this one, and this one. That means dominant for smooth, but recessive for color. We find that there's 3 of those. If we look at the opposite of that, where it's double recessive for smooth, in other words it's wrinkled, and dominant for color, then we know that should be a straight line there. Then there's 3 of those as well. There's this one, there's this one, and there's this one. So that's 3. Big little s, big Y something. And then finally there's one which is double recessive for both of them. So that gives us our phenotypic ratio of 9:3:3:1. And that's the classic number that you get when you do a dihybrid cross with a heterozygote. That's this guy over here. So let's cast our mind back to meiosis, and where we had the random assortment of chromosomes in metaphase 1 and 2. And this random assortment at metaphase gives us equal combinations of chromosomes and gametes. And this is where Mendel's second law of independent assortment comes about. Let's have a look back here. Here is a cell lined up in metaphase 1. And if you look here, we find that the dominant A genes are on the same side of the cell as the dominant B genes. So when they separate, when they go left and right, we find that the dominant A's and the dominant B's go to the same side of the cell. And consequently, the recessive A's and the recessive B's go to the other side of the cell. What this means is that when we split this up in meiosis 2, we have a dominant A in the same gamete as a dominant B, and we have a recessive A in the same gamete as a recessive B. So this gives us gametes of big A, big B, and little a, little b. The alternative arrangement at metaphase 1 is that the chromosome with the dominant A's happens to be on the same side of the cell as the chromosome with the recessive B's. So these two have swapped places, these B chromosomes have swapped places in relation to the A chromosomes. And it's a purely random event, so it's 50% do this and 50% do that. And so when this goes through meiosis II, dominant A's and the recessive B's are on the same side of the cell, we end up with the dominant A and the recessive B in the same gamete, and the recessive A and the dominant B in the same gamete. So we can add those to the mix of our alleles. We have dominant A, recessive B, and recessive A, dominant B, and these are our 4 possible combinations of gametes. So these are how we get 4 possible different combinations of gametes going into the next generation by this independent assortment in metaphase 1 giving us this combination here or this combination over here. It's completely 50-50 whether it happens or not. What we can do is we can take these alleles, these gametes, and put them onto a Punnett square. We see that in the next slide. So we're back to the S's and the Y's, but here are double dominant, one dominant, one recessive, the first one recessive, the second one dominant, and double recessive, and we can fill these out here. And if we see here, this is the same diagram but a much neater than we did a couple of slides ago. Here's our one green wrinkled double recessive seed, and across the top and down the side and up the middle are the 9 double dominants, and then 3, 1, 2, 3 dominant smooth recessive color, and then 1, 2, 3 recessive smooth and dominant color. Mendel's second law, his law of independent assortment, says that segregation by one pair of alleles, in other words the A's, does not affect segregation by any other pair of alleles, in other words the B's. So you can do what you like with one and it doesn't affect the other, and the reason for this is it's on different chromosomes. As long as those 2 different pairs of alleles are on different chromosomes, then Mendel's second law is true. So Mendel's laws of inheritance, his principle of segregation and independent assortment, they're very good fundamental principles of genetics, but they only work when we're talking about single genes, such as A's and B's, or S's and Y's, that have a standard predictable dominant or recessive characteristics. And there are many genes that do not have these particular traits. So we're going to have a look at some exceptions to the Mendelian laws of inheritance. One example here is incomplete dominance, and then there's also codominance and multiple alleles and epistasis. These are some 4 examples that we're going to look at. So let's look at incomplete dominance. Normally you would expect a dominant gene combined with anything else to be the dominant phenotype. And in the case of incomplete dominance, that's not the case. What we have here is, if we have big R in this case, we have a red petal. That's this one right here, big R. And if we have a double recessive, little r, then there's no pigment, and so you have a white petal, and that's what you have here. The incomplete dominance is where we have a heterozygote, which is this bottom diagram here, and we have pink flowers. And what you can think of here is that each dominant allele makes a particular amount of red protein that makes those petals red. If you've got double recessive, it doesn't make any red protein. If you have just one dominant allele, it makes a little bit of red protein. In other words, it becomes pink and not red. So that's incomplete dominance. Double recessive is white, double dominant is red, but heterozygotes are halfway between the two, and in this case, it's pink. Another exception to Mendel's laws is if you have multiple alleles. In other words, if you have more than 2 alleles. So if you have more than 2 alleles, this is what we're talking about here. And in this case, we're talking about blood types. We have 3 different possible alleles here. They're all represented by the capital I, superscript A in this case, superscript B in that case, and then we have the recessive version, which is lowercase I. So here we have 3 alleles. This one codes for the A protein, this is for the B protein, and this one codes for no protein at all. This is recessive. Because these 2 are still dominant, And because, as you can see here, you can have a genotype that has both big A and big B together, causing, in this case, type AB blood, not only do blood types display multiple alleles, they also display something called codominance. So codominance is where you have expression of both members of a heterozygous gene pair. So in this case, you can have a genotype that looks like this, and this produces the A protein, this produces the B protein, and so both are dominant. We have codominance between these different alleles. Neither one of them are recessive. In the case of the recessive type, such as say here, you would have big I, big B, and that would produce the B protein, but the little I, or the big I-O, produces no protein. So in this particular diagram here, little I is recessive, and it's also known as big I-O. It depends on the textbook you're looking at. Little I and big I-O are the recessive trait, the recessive allele of the blood types.